my name's again Corey Giles. I'm a postdoc at Oklahoma Medical Research Foundation in the Jonathan Wren Lab. And so unusually for people on our campus, at least, I am 100% computational and statistical. I have done some wet lab experiments, but for the most part, our lab has two focuses. Number one is that we help participate in collaborations, analyzing other people's data, and there's an enormous amount of DNA and RNA sequencing experiments, various other types of high throughput experiments, and then we also, our primary research focus is in the general area of taking publicly available data sets in the molecular biology field and either using that data to derive new insights about biology through meta-analyses and machine learning and that sort of thing, or to be able to put data from individual collaborators' experiments into a broader context, so they can ask questions like, well, we saw X, Y, and Z in our experiment. Have other people seen that? Or have other people seen these two genes go up together? Or we also can answer some more open-ended questions like, what does this gene do? Can we guess what this gene will do, even though nothing has been published about it yet? Which is a pretty common scenario in biomedical research. Even for humans and mice, which are the most heavily studied species, only about 2/3 of genes have ever been looked at by anyone. And it's much less, specifically, and it's much less common as you go down to other less frequently studied organisms like worms and flies and dogs, so on. Okay, so what is definitely the case in molecular biology, and it seems to be generally the case in most other fields, is that the number of, so this is a log scale graph showing how many papers have been published in PubMed over the last 20 years. And the amount of publications are growing exponentially. The number of data sets is growing exponentially, even though, so these are all different methods to quantify either DNA or RNA, various modifications of the levels of different RNA transcripts. And each one of the colors in these graphs is a new technology. So the technologies change, but the amount of data being generated continues to grow exponentially without too much indication that it's going to be slowing down. And this is both a difficulty in the sense that it raises it greatly increases the amount of complexity involved in managing all of this data and organizing it and storing it and processing it. But it's also an opportunity in the sense that, we in our lab, at least, and many other labs, see it as an opportunity to reuse the data for purposes that the original investigators may never have gathered it to do. For example, my dissertation was on quantifying, and I'll talk a little bit about it, quantifying age-related gene expression changes. And to do that, I used tens of thousands, actually hundreds of thousands of samples that had been gathered for all sorts of purposes by different people. But when they labeled those data sets with the age of the sample, I was able to use it anyway for a purpose that they never originally intended. So that's just one of many uses of public data sets, and we'll be talking a lot about public data sets and sort of the relationship between the individual researcher and those public data repositories, which are certainly field specific. So that, yeah, as I was just saying, there's a variety of different applications of public data. Many people, I don't know, people have a range of attitudes towards, you know, in biomedicine, we're in many cases mandated by the NIH to submit certain types of data that we generate to public repositories that they have developed for the purpose. The Gene Expression Omnibus, the Sequence Read Archive, the Protein Data Bank. If you accept NIH funding, you're required to submit those data upon publication, at or before publication. And the NIH wants to put a strong focus on that for a variety of different reasons, so that people can reuse it for machine learning, can reuse it for meta-analyses like I was just talking about, or reanalyses, which would be, you know, if someone wants to take your data and try to crunch the numbers themselves and see if they come to the same conclusion, that would be a reanalysis. But I think the most important purpose of it is the general is enabling general scientific reproducibility. We'll talk about that. The scientific reproducibility is an incredibly complicated topic, but you can't even begin to reproduce something if the data is not available. So here's a couple of examples that either are famous in my field or that we did in our lab. Okay, so this figure up here is one of the most influential figures in the aging field in the last decade or so. It's called the epigenetic aging clock. It was developed by Steve Horvath at UCLA, and so the background for it is that for a long time people have been looking for a biomarker of age, meaning something that we can measure in the body that can predict really well how old you are. And there's a variety of different reasons why we might want to do that, ranging from, you know, the most important one is probably that if we have a good one, we can, you know, in the aging field, one of the things that we want to do is find drugs or interventions that improve lifespan or health span. So, but those experiments take a very long time. Even in mice, they take years. So if we have a good biomarker, we can maybe narrow down our candidates before we perform the full experiment. And this is, it's a very hyped thing now. Actually, several applications based on this epigenetic clock have been commercialized. I think that there's at least one insurance company that will let you find out what your epigenetic age is and use that to adjust your premiums, because another application of it is that, this is controversial in the field, but if your, you know, epigenetic age is older than your what we call chronological age, then you might be at higher risk of a variety of age-associated diseases. So, you know, it's a useful research tool, and whether or not it should be used yet in the clinic or given to people, I don't know. But the point of here is that this clock, what he did was take tens of thousands of publicly available DNA methylation data sets, which were labeled with age, and then trained a machine learning model to predict the age based on that data. DNA methylation is a chemical adduct that goes on top of your DNA. It doesn't change the DNA itself, but it's You could think of it as well, I don't know, chemical additions that can be added or removed. They're not permanent. They're often used to turn on or off genes. So this was a high-profile case of reusing public data to make an advance in a field. And like I was saying, for my dissertation, used publicly available data to, the purpose of it was to find genes that change their expression levels, their RNA expression levels, consistently up or down with age. And the purpose of that was to sort of possibly narrow down pathways or genes that might be important in aging. And that too was dependent on having this data available. Another application in our lab is called GAMMA. And what it does is predicts the functions of unknown genes that have not been studied before in the literature, using a concept called guilt by association. So taking the gene you want to know about, but has no literature on it, and then finding which genes it shares regulation with according to this public data. So this data comes from public gene expression data, and then we can identify what these known genes do based on what they're associated with in the biomedical literature. And our literature database is called PubMed. So we're using two separate open databases here to infer the function of unknown genes, and we've validated that this works, and have actually published in collaboration with people. The way we often use it is that we have a collaborator that's interested in cell cycle or some specific topic, and they say, can you find me a gene that's never been studied before that's related to my thing of interest? So all of that's enabled by public data sets. Okay, but let's talk a little bit about scientific reproducibility. And I'm glad we may have a psychologist here, because it's a topic that's near and dear to my heart, because when I was doing my this meta-analysis of age-associated gene expression changes, I was disappointed to find that there wasn't nearly as much consistency as I was expecting or hoping there to be among different experiments and what they said about which genes change with age. So the reproducibility crisis exists in many fields. This is a study called the Reproducibility Project Cancer Biology, which took 193 experiments from 53 papers, and these were all high impact, big shot papers that made a big splash in their field. And they attempted, and basically all the project did was take all of these findings and attempt to do exactly what the paper did, and then to find out whether or not they could reproduce the results. And and this was in, the papers were published in 2010, 2012, which is before there were as many mandates for open data as there is now. So 2% of the experiments they tried to reproduce had the open data in the first place. For 70% of the experiments they tried to reproduce, they couldn't buy the reagents. They had to ask somebody who had them in a freezer somewhere to send them to them. And then of those, only 7 out of 10 were willing to share those reagents. None of these experiments were described in sufficient detail for them to be able, just based on the paper's methods alone, to reproduce each aspect of the experiment. 32% of the original authors were not helpful or not responsive, and 41% of the original authors were very helpful. So that's good. And then in some cases, it turns out that the protocols may have been incorrectly described. They didn't actually match up exactly with what was done in the experiments. So this we're not even. In my first paper, I learned the grammatics approach. Somebody is trying to talk or ask a question, but it's very echoey. Is that just me, or I can't understand?
I don't think it was the question. I think you're okay.
Okay, just somebody. Okay. So this is before we even start trying to get into the results of what was reproduced and what was not. This is just the challenges in doing the reproductions to begin with. Then here's the the summary of the findings is that on average, the effect sizes of the replication were 85% smaller than the original findings. 40% of effects replicated on more successfully on more criteria than they failed. And positive results were half as likely to replicate successfully than original null results, which should not be surprising if you know anything about statistics. And so this is this is a problem. And this was one of the impetuses behind a lot of the reproducibility initiatives. And so, I think it's going to be a while before the scientific community, and I know for a fact it's not just biomedicine that has this problem, but we certainly do have it, a while before we fix it in its entirety, because there's a lot of moving pieces and reasons why reproducibility is not as good as it could be. So John Ioannidis is a well-known researcher in biomedicine that studies reproducibility related topics. And this paper, what does research reproducibility mean? I think it offers a really helpful conceptual schematic for what are the different parts and pieces and types of reproducibility. So this figure on the left is from the paper, and each one of these colors is a different field. So a different field of study ranging from physics to agricultural sciences to medical research and economics. It's very broad. And so what he's showing here is that the number, the absolute number and the relative percentage of papers being published related to reproducibility is increasing in nearly all these fields. I guess there are a few where it's decreasing with time, showing that there's increased relative and absolute interest in these topics, probably because people are it can be very difficult if you base your research on somebody else's paper and then you find out that you can't reproduce what they did. That can waste a lot of time and money. So he breaks it down into a couple different types of reproducibility, because we use that word, but it doesn't. There's many different things that it could mean. So actually reproducibility, the word, I was surprised to learn from this paper that it actually entered the lexicon from computer science. What it meant in the computer science world was that if you run the same program with the same data, you should get the same output. And that's certainly true, but that's a very restrictive definition compared to science as a whole. So methods reproducibility would mean that if I read the methods section of your paper, along with any supplementary materials that you provide, then I should be able to do what you did. And it should be described in sufficient detail to where if I try to reproduce it, I will end up doing the same thing, which is very difficult in many situations, given how short method sections often are compared to the reality of how much work was done. Although, this is getting a little better because now things are being published online and there's not as much space limitations on supplementary materials. Now down here, this is where it starts to get more interesting. Results reproducibility means that if I did the same thing that you did, including both the experimental aspect and the analytical or computational or statistical aspect, that I should get the same general results. Obviously not exactly the same, because there will be statistical variability always, but I should get some result near in the ballpark if I did the exact same thing. Robustness and generalizability takes it one step further, and it would, in my field, it would involve something like, okay, if I give a certain drug to a C57 Black 6 mouse, and I observe X, Y, and Z, then I should see X, Y, and Z. I may or may not find X, Y, and Z in a different strain of mouse. That's, that is not necessarily something that has to be true all the time, because there could be differences between the mice that legitimately cause a different effect. But having robust and generalizable results increases the confidence that your finding is really significant and important. In human medical research, we would prefer to have drugs or findings that don't vary a lot between people ultimately. And then finally, inferential reproducibility is sort of at the highest level of the conceptual pyramid here. It talks about, is the result true? Are the conclusions that you drew using your data analysis experiment, are those broad conclusions true, or would they be reproducible? In other words, are they not, extremely dependent on the exact specific details of your exact experimental protocol, but would they be, true in a more, maybe popular level sense? Like, if I'm measuring cognitive performance and I say my drug improves cognitive performance, then that shouldn't depend too heavily on which particular measure of cognitive performance that I use. So these are a lot of different types of reproducibility, and it's worth keeping in mind which one we're talking about in a given moment. Okay, and there's a variety of different reasons people that reproducibility can go wrong, quite apart from anything computational. But I think most people know about p-hacking. It's also, or harking is something related, where it and frankly I see it all the time. It means hypothesizing after the results are known. So you do an experiment, maybe that measures a lot of different things, see which things changed, and then you say that was your hypothesis to begin with. So that increases the researcher degrees of freedom, and it increases the risk of false positives and irreproducible results. So does low statistical power, which is very common too in my field, because experiments are very expensive. People try to get away with, as few mice or whatever as they can. And then publication bias is another big issue, where sometimes one manifestation of it is called the file drawer effect, where people do not publish if they didn't experiment and didn't see a result. They won't publish, and they'll only publish if they saw something. And that could result in a situation where 10 people didn't experiment, only one of them saw a positive result, and that's the only thing that gets in the literature. So it looks like it's true 100% of the time instead of 10% of the time. I think, and these topics are covered in RCR courses and so forth. I think that one thing, a lot of people talk about these issues as if, like, yeah, I know about them. But I really think one of the most interesting people to read on this topic his name is Andrew Gelman. He's a statistician. He has a really fascinating blog down here, and he wrote one of the papers that he's written on this topic is called The Garden of Forking Paths. So in that paper, he makes the point that first of all, p-hacking does not have to be done consciously. In fact, if you think about it, if you have a broad general hypothesis, there's potentially hundreds or thousands of different ways that you could run the experiment and analyze the data. This mostly comes into play when you're analyzing data. In the case of a sequencing experiment, there are, dozens of different aligners, dozens of different normalization methods and differential expression callers. And so there's thousands of ways to analyze the same data set, actually. And his point is that, a lot of I've seen people do it not infrequently, that, they try one way, it doesn't work, and then they try something else and they keep trying it until it works. Well, by which they mean it returns some result that they consider acceptable. Well, they may not realize that they're p-hacking there. In fact, they probably don't. It's unintentional because they just think, because like experiments don't work if you don't sometimes. If they don't work, try them again. The solution to this is to pre-register, not necessarily formally, but you need to know in advance what you're going to do with your data before you do it. Okay, so in summary, on reproducibility, it's a huge problem, fundamental to scientific endeavor. There's examples in multiple fields, and this is called the power pose. I don't know if we have our psychologist here, but this is one of the ones that set off the general. I mean, the to their credit, it was the field of psychology that really first sounded the alarm on these reproducibility issues. Because they're the ones who are always thinking about cognitive biases and so forth. So, and they found out that a lot of their own literature was not reproducible, and we scoffed at them and said, oh, well, that's because they're soft science. And then we found out, oh, no, it's exactly the same in biology. Okay, but so this may have been too much of a tangent, but the point is we're going to talk about like data integrity and public data and stuff. And my point is that, the reason, the fundamental, most important reason why we care is because we want to improve reproducibility. And one small aspect of that is to make sure that we manage our data well, take care of it, submit it publicly, and a couple of things like that we'll talk about. Okay, so the formal, one schema framework set of principles that's being promoted by the NIH is called FAIR. It's just a list of guidelines or things to think about that you should do to make sure that your data is publicly available in a way that other people can use, for all these reasons that we were just talking about. So is it findable? Meaning, can somebody else search for your experiment that's relevant to them, and can they find it based on some reasonable method? Is it accessible? Meaning, can they actually download the data once they want it? Meaning that it doesn't count if you keep the data, but you just keep it on your own laptop or on your external hard drive. Even if they know it's there, it's not accessible if they can't download it without, begging you over email to send it to them. Interoperable refers to both, a secondary goal, I should say, also of these FAIR principles is not just to make sure that other people, humans, can access and reuse this data, but it's also for computers, and which is increasingly important because of machine learning and meta-analyses and all the things that I was talking about. So their goal is to make it both human and machine accessible. So interoperable, that refers to using whatever file formats is standard in your field for your particular type of data, not some esoteric format. And then also, to the extent possible, linking the metadata, meaning the descriptions of what were these samples that you collected, and what did you do to them, and so forth. It's easier for computers to understand that if you put it into some structured format. I'll give an example in a second. And then, is the data reusable? Meaning, is it complete? And in terms of both the data and the metadata, and do you describe where it came from, what was done to it, and it doesn't count if they have to like if it's not openly licensed to where they can legally use it. Okay, what do I mean by standardized metadata? In my field, we have a set of what are called ontologies in the obofoundry.org, which basically means assigning an ID number to various terms. The one non-medical people may be familiar with is ICD, which is used by doctors. Whenever you go to the doctor and you get a diagnosis, they give your disease, whatever you're diagnosed with, an ICD code, and that's what goes to the insurer and so on. But we have cell type ontology and gene ontology and all different kinds of things like that. So instead of using words, you use an ID number, and the reason is because oftentimes the same concept can have many different synonyms that people can write different ways and so on. And it's hard for computers, or hard for people, and even harder for computers to know that people are maybe referring to the same concept with different words. So I wrote a paper trying to do this on one particular topic, which was to go through gene expression data sets and turn, so this is what the raw data looks like in this gene expression omnibus database. People put in a series of key value tags that are not structured or standardized. So for example, somebody could write sex male, sex M, sex 0, or sex 1 actually. Or they could, there's a variety of different ways they could describe the sex. It's not controlled, it's not structured. In order for the computer to use it, you have to make it structured. Similarly for age, 32 what? That's a particular problem with mice, where it could be days, weeks, months, or years. And so this problem is non-trivial to solve after the fact. So it's always helpful if people who are submitting their data do that to the extent possible. Now this is a little bit about how, broad steps of how you could take your data set and make it fair. Or how you could take any data and make it conform to these principles. So you analyze it, you define a semantic model, you make it linkable, which primarily refers to using IDs to link samples and experiments and perturbations together. In other words, you got to have a unique ID for each of the objects that your data consists of. Assign a license, define the metadata, and deploy the data resource, which means, in practice usually means submitting it to a public repository that's already created for your type of data. So this is more for people who want to do this on a large scale. So you can go to this, let's see here. You can go to gofair.org where they will explain all of this, but there are various implementation networks. Like these principles, it's not just biology that's trying to do this. I mean, it's at least, materials, science, chemistry, ecology. I don't know how, physics or psychology, I couldn't tell you, to be honest. But and maybe they have a different paradigm in those fields. Although I wouldn't imagine that the principles themselves would be wildly different. So basically, if you're interested in doing this, you can go to this website. You can find out if there's already a community that's doing this in your area, and then join up with it and ask them for further instructions or information that's specific to your field, because this will differ from field to field, how exactly it's done, which metadata ontologies and so forth need to be used, and where the repositories are, what file formats. It's all field dependent. Okay, so in summary, it's a broad, high-level framework for how to tackle this particular aspect of the reproducibility crisis, which is, making just the data itself publicly usable and reusable. That's only one component of what will be needed, but it's a start. And so at a broader level, there's this concept of data stewardship, which I guess the point of this figure is to sort of let you know that I know this stuff, it sounds bureaucratic, jargony, and time-consuming, and that people aren't going to be able to do something like this overnight, even if I persuaded you that it's worthwhile. So you can take a step-by-step approach, just slowly building up over time, starting with what we'll talk about next, which is something as simple as just making sure you don't lose your data, which happens more often than you would think. And then, using unique IDs for your samples and linking the data and, keeping your data and metadata together, which is another common failure mode. Then you can add searching on it. Then you can, use probably concurrently use methods to make sure that the analysis, the code and so forth that you run on your data can also be reproduced. And then, once you have all these pieces together, you're most of the way to FAIR. Okay, so this these are some what I'm going to be telling you next. It's not something that most people are unaware of. It's just they may not be. It may not be something people always put into practice. So I summarize it as you need to make sure you don't lose your data. Okay, so at a basic level, at the most basic level, you should be backing up your data, and everyone knows that, but not nearly everyone does it. And so, this was a incident that made international news from OU HSC a couple of years ago, where a researcher had the only copy of 3 years worth of their cancer research data on their laptop. It got stolen out of their car at Panera, and they had to go to and publicly beg for whoever stole it to give it back to them. And they have to offer a reward. And I mean, Oklahoma got made fun of big time, in the United Kingdom and in Europe, continental Europe, and so on for that. And but that could happen to a lot of people, because backups, if you don't have an automated system for doing it, then you'll forget, is the point. And then the point of this figure on the left, this comes from one of our PIs. Some people consider this sort of setup to be a backup. Well, where they have their data in the on external hard drives in their joint backup / liquor cabinet. Well, there are better approaches. Okay. Now, another reason to want to back up is that people don't often know this, don't think about it until it happens to them, but hard drives break. They wear out over time. They have a defined lifetime, And the longer you wait, eventually your hard drive will break. In fact, after I gave a version of this talk in January, my hard drives on my desktop broke like a couple weeks after. I thought that was really timely. And fortunately, I had been taking my own advice and I had backups, but and I had automated backups. But it does happen. And also, there's some once in a while there's hard drive batches that are manufactured that are defective. Like the whole model is defective. So this one here had a substantially higher failure rate than the rest. So even if you've had you never had a hard drive break on you before because you always cycled out your laptop in time, you might get a bad batch next time. My boss also ordered a laptop within the last month, and it broke within 2 weeks of him getting it. So it can happen. And it doesn't matter also very much whether you're using the hard drive or not. It has the rate of failure has more to do with the amount of time rather than the amount of usage. So, it certainly a drive will live longer if it's usually sitting in a drawer, but still not infinitely. And also, external hard drives are really prone to shock damage because they're just they're made of spinning plates. If you jostle them, it can seriously damage them more easily than for other types of hard drives. Yeah. So this is what a hard drive looks like inside. It's got a spindle that reads and writes from this rapidly spinning disk, and so you can clearly see if you jostle it, you're at risk of a problem. And the reason I don't know about you guys, but I know that this is absolutely common practice at OHSC. People store data all the time on external hard drives, and they think that it's a suitable backup. Maybe in your field, people know better, in which case, good for you. Okay, so a better paradigm is to use a combination of local and global redundancy and an automated approach to sync them together. So local redundancy, it means something like RAID, which I'll describe in a second, but it basically means that you can install you can install multiple hard drives in your computer in such a way that it will keep each bit of data on more than one hard drive simultaneously, so that if any one hard drive fails, you are out of luck. This is not a backup solution, but also, in addition to that, it's a way to make sure that if a hard drive does fail right before your grant deadline or paper deadline, that you can keep going and you don't have to scramble. You'll still have to replace that hard drive, but you'll still be able to keep going for a while. And then global redundancy, people have gotten a lot better about this, but basically it means storing it in the cloud or storing it at your institution's data center solution, whatever that might be, because they typically take this type of stuff a lot more seriously, and will have offsite backups and data stored in multiple geographical locations in case anyone burns down or gets hit by a tornado and so forth. And then an automated approach to sync A to B, and that depends on your platform. So there's Apple. I use Apple. I don't remember the name of it, but it's got both Windows and Macs have a. I don't know about Windows either, actually, but I know Apple has an automated backup program. Time Machine, that's what it is. Although again, I think that's largely intended for externals. This just describes what RAID looks like in case you haven't heard of it. So this is just pointing out that each one of these columns is a disk, and so A1 and A3, just pointing out that each segment of data is replicated on more than one drive. And this can be somewhat of a hassle to set up for yourself if you don't know how, but you can buy machines from Dell with RAID already set up and installed for you. And probably your IT services will be able to tell you how to do it also if you don't know how. This is, yeah, I'm just going to leave this here for a second. This is how to do it on Linux. If you want to read this, you can look at the recording. But the point is, it's not actually that hard. Okay. Remote cloud storage, Dropbox, Google Drive, Amazon Web Services. Yeah. So the main point here being that remote backups, remote storage is good, but you should think of it. The difficulty with something like mounting a network drive is that it can drastically slow down your computations and so on whenever you're using it. So you should think of the network storage usually as being a backup rather than a primary data storage location. And then on Linux, you can set up a cron job, which is like a repeatable service that will run every so often however often that you set it to, and it will run any command that you want. And you can just run the rsync command. Like this is a very, very like simplistic poor man's automated backup script. And there's better and more complicated ways to do it, but this is, way better than just backing up whenever you think of it, because you'll inevitably forget whenever it's most inconvenient to do so. Another thing to think about when it comes to just simple data integrity is to keep in mind who is responsible for it, because sometimes research projects involve multiple people spread across multiple labs even, and everyone thinks that it's everyone else's job to keep the data, and then no one does sometimes. And so this happens to us a lot. We analyze data that's generated by other labs, and occasionally people will come back 3 years later or 5 years later and they say, hey, do you still have our data? Because they sent it to us to analyze. We analyzed it, sent them back the results, and then they publish the paper and they think that we just keep it around forever. Well, broadly speaking, if you're the one who generated the data, it's you or your boss's job to not lose it. And it's also something to think about too, whenever you've got graduate students coming in and out of a lab, that probably it's the PI's job ultimately, not to rely on a graduate student or even a postdoc. At the very least, they should have a plan to ensure continuity. In the case of sequencing specific data, the point of this is that we can upload data to NCBI, the National Institutes of Health, before the experiment's even done. So once it's there, it's pretty much safe. They'll take care of it, and you don't have to wait until the paper is published to do that. Okay, whenever it comes to linking together data and metadata, this is a complicated topic, and I just want to but basically this is the most common mistake that I see, is that metadata is not put into normal form. What does normal form mean? Basically, it means that if you've got a spreadsheet full of samples, well, what it means is that you actually have typically a hierarchical level of organization of different objects in an experiment, ranging from a sample. You could have an experiment consists of multiple experimental groups or perturbations, which each one consists of a sample, and then those samples might even, in the case of a mouse, you could have three different tissues extracted from the same mouse, and so on. And so you should have a different table for each one of those layers of the hierarchy. This makes statistics easier. This makes data submission easier, and the way that you can typically tell if you've got a problem is that if you have things that are being duplicated across multiple rows, that's often an indication that you need a separate table, and then you also would, for every layer, you want to have some sort of unique numeric ID for that particular sample or sample tissue combination or whatever it might be. So this will be obvious to people who have worked with databases, but it's not so obvious to others. I think these are examples of what that means in the biomedical context, and since we're not all biologists here, it would differ for each field. But I think something that non-analysts don't often think about is to record batch effects, meaning which technician collected the sample, what day was the sample collected, things that are related to how and when and who and where the data was collected, rather than the data itself or things directly related to the sample. So if you record those things, you can often actually end up making better use of your data by correcting for batch effects, which it's not uncommon that mice, for example, will react slightly differently to different technicians. Maybe, I don't know, if they smell different, or if they're more scared of a tall and big guy, or what. But it's a known phenomenon. And sample IDs, I think this would be true across fields as well, is that you don't want to have a sample ID doesn't tell you anything about the sample. It's just an ID. You keep the any data that's descriptive about the sample in the metadata table, not in the name or file name or what have you of the sample itself. And then, so all of this is what metadata means and consists of, and it should be stored with the data, which doesn't always happen. If somebody goes back later and they find the data, they don't have the metadata, it's useless. When it comes to computational reproducibility, I'm a big fan of this paradigm that comes from a service called CodeOcean, and it doesn't. The main point here, you can check it out if you want. The main point here is not that you should use CodeOcean. That will depend on your what your field is. But I think what would be true across all fields is that a computational analysis consists of three components. It consists of whatever code you're running to analyze the data or generate the figures or what have you, the data itself, and the environment, which means all the packages, all the specific versions of the specific code packages that you're using to run the analysis, because things can change from version to version. And without having all three of those together, you won't be able to fully reproduce the analysis. In my experience, keeping the environment is the one that's most often a problem for people. And how to do that will depend to some extent on which programming language you're using. In biology, it's pretty much universally R or Python. But the point is, you need to use your language's specific functionality to at least record the versions of the packages that were used. And oftentimes it doesn't hurt to put the major ones in the method section of the paper either. Yeah, so this is what CodeOcean looks like. And you can see over here that it's got, it stores the environment, the metadata, the code, and the data all together in the same interface. And there's a variety of different ways to do this, but the point is just all of those things need to be together in whatever solution you choose. Docker solves a lot of these problems too, and CodeOcean is based on Docker. When it comes to code versioning and storage itself, you might not. A lot of people, when they're editing code, they're new to editing code, they just every time they do a new major analysis, they'll save like another copy of the file, like my analysis version _v1, my analysis _v2, which is better than nothing. It's certainly better than just like editing the file and then forgetting that you changed it and relying on the old results. But I think everyone who codes knows to use Git and to use an external Git repository to save your code. But if you don't, it's not too hard to learn the basics. This is one website where you can learn it. And another thing is to keep in mind that there are code-like parts of an analysis that are not necessarily code themselves, but are, this might be too specific, but an example would be, I guess, like in meteorology, if you're running an analysis that relies on a certain geographical database that is improved over time, you need to make sure to not just say we use that database, but which version of that database did you use? Even though it's not part of your data, it's not part of your code, but your analysis depends on it, and it can change. If those two things are true, you need to record that. And ideally, record it in a way that your analysis will be able to automatically reproduce it. Pip freeze, Conda, virtual environment are the solutions for Python version manage or package version management, but you can also solve a lot of this problem by just using Docker, which creates, it's sort of like a recipe for building a virtual machine that has a specific versions of all the packages, and people can be sure that when you're using it, that they'll have the exact same versions of everything that you did. Okay, that's sometimes in your field there may be like third-party websites where you can do your analysis there, and you can save them and so on, and those are fine if you could be sure that they'll be there 5 or 10 years from now. That's one thing to keep in mind. And the downside of using things like that is that it's difficult to really know exactly fully what they're doing, because they're often GUI tools. And a lot of times commercial software packages will, you can't version control them. Like you can't always roll back a commercial software package to the exact version that you used at the time. So it's limiting in that way also. So broad, big picture on analytical code reproducibility is this. There's a lot of ways to do it. It depends on your language, depends on all that. But you can just test whether it's happening or not for you. Is a simple test to know if everything is okay in this regard is if you can basically get 2 different people to reconstruct an analysis by pulling the data, put them in two different rooms on two different computers, give them the basic instructions, and if they can pull the data, set up the environment, run the code, and get the same answers, then your analysis is pretty much reproducible. And you'd be surprised, whenever we try this in our lab, the most common way it can fail is if people find out that, like, some part breaks, like they get an error because they're installing something incorrectly or what have you. And that's an indication that this process isn't automated enough. Okay, so we've kind of covered a gamut of high-level topics here, but in summary, the reason why we care about data stewardship, data integrity, public data sets, FAIR principles, all this stuff is because of reproducibility above all. And there are many different aspects to it. No one's perfect. Few of us have the resources to do all of this perfectly. But I'd recommend taking it, step by step. So with that, here's a picture of our lab. Thank you, and please ask if you have any questions.
Thank you, Corey. A lot of stuff there that if you're not familiar with it, we do teach classes or workshops on a lot of aspects of the things that Corey has mentioned. And please, if you have any questions, you are welcome to either unmute or to put it in the chat.